Welcome to Sigest, your fortnightly serving of digestible science from plant and food research.
Kia ora, hello, and welcome to another episode of Sigest. My name is Tom Moore. Moving from science to management is a major decision and is not always successful. In this podcast, we speak to Dr. Jolon Dyer, Group General Manager, Science Services, about his science journey so far, including the challenges, rewards, and learnings with moving into management from a successful science career. Jolon, welcome to the podcast.
Thank you.
Thank you so much for being with us here today. This is A Science Life for Me, which is about your journey into science and then into management. So would you first be able to give us a point in time that you decided that science was right for you and started to pursue a science career?
Well, I had a very unusual entrance into science. Right throughout my schooling, I always liked every single subject. And when I first enrolled to go to university, I was actually thinking more of doing an arts-based major.
And what sort of arts were you interested in doing?
I was very interested in English literature, and also at that stage I was studying French language and literature.
Okay. So was that a point where you had to decide between going down the arts route or going down the science route? Or was there a particular person that encouraged you to go a certain direction?
Yeah. What happened was, so I'd enrolled, I still hadn't decided what I really wanted to do. And so in my first year, I'd enrolled with a whole range of courses, including chemistry and maths and English literature and French, etc. Then on the matriculation day, this is the University of Canterbury, it had on the matriculation form, please go and see a certain professor in the chemistry department. So I went to see this professor and had a discussion with him, and he invited me to join honours chemistry course, which had the very attractive aspect that you skipped stage 1 completely and went straight into stage 2 courses. And I thought about that, and that's actually how I ended up going the science direction instead of arts.
Interesting. Have you managed to keep the arts in a recreational capacity alongside the science?
Yes, I got permission. The honours chemistry course was very intense, but I got permission to continue studying French as well while I was doing chemistry. I also, partway through my PhD, I started learning Japanese as a hobby, and then I got very passionate about that, so I ended up studying Japanese as well.
So you grew up in New Zealand, and learning these languages, did you have the intent to go overseas at some point in your life and live within the culture of those places?
Initially, no. So from a very working class background, didn't really have any opportunity to travel overseas until a little bit later in life. I learnt the languages because I really enjoyed communicating with people from different countries that were visiting New Zealand, and just really enjoyed that ability to be able to communicate in a different language and the kind of smoothing of the communication you get with that.
I agree. It's a really good way to get into and understand others' cultures. And at high school, university, it's kind of a melting pot of different cultures and backgrounds, and it's just fascinating to be able to engage more with those stories. What particular was it about chemistry that drew your attention? So when I think of ecology, you've got these macro things you can physically touch and move. I always found chemistry quite difficult because you can't see the sort of things that are going on. Was there like a particular thing that you kind of drew you to chemistry?
Yes, I really like abstract concepts, so maybe quite the opposite of what you've just described. So I've always liked music, languages, which have a lot of abstract things inherently within the communication. And chemistry, I really loved the concept of the molecules and how the molecules come together. So even though they're at a scale where you can't see them, just that conceptualization of how they would come together, particularly biomolecules.
Interesting. Quickly, what music did you play or perform?
I initially learnt the violin and also did piano through to grade 7, and also taught myself a little bit of guitar, and used to be in a very minor band with my brothers.
Wonderful. That's fantastic. So when you say about how molecules are interconnected, was that part of a general kind of ethos about how lots of things are interconnected? Like science, for example, is linked with arts, is linked with these kind of general overarching concepts. It sounds like you maybe were quite academic in your younger days. So could you speak a bit about that kind of process a bit more?
Yes, I was very, very shy growing up. And so when you mention being academic, that's probably a fair comment. I was often called a nerd at school. I was very, very shy, very introverted. I had a stutter on and off from about age 12ish to about maybe 21 or so. Kind of on and off. It wasn't too bad at some stages. But I think what that made me do is I ended up getting very immersed in the subjects I was studying. And you've mentioned the interconnectedness of things. I always enjoyed that really holistic type thinking, and truly believe that things are kind of connected to each other. So yeah, the way you've described it's actually very good.
Interesting. Going through your studies and how it kind of progressed, did you perform a master's before going to a PhD, or honours then PhD?
No, I was able to go straight through to honours, and then from honours directly through to PhD. So got first class honours, and if you get first class honours, usually able to be offered a PhD directly.
And that was offered at the same university, the University of Canterbury?
Yes.
And how many years ago was that now?
Good question. This was in the 90s.
Okay. And could you speak a bit more about what the content was of your PhD?
So my PhD was in synthetic organic chemistry applied in a medical application. So it was around Deal's order approaches to anti-cancer prodrugs. So
you may need to expand on that a bit.
So essentially synthetic organic chemistry is where you design molecules that don't exist essentially, and you work out synthetic pathways by which you might be able to make these. So with bowel cancer, which is a very bad problem here in Aotearoa, within the growing tumour, you end up with a deficiency in oxygen. So there's not enough blood and therefore oxygen to the growing area. And so that we targeted creating molecules which essentially had a bridge type structure, and so they'd be relatively harmless, but when they came into a low oxygen environment such as in the tumour, the bridge would snap open and they'd become an intercalating agent. An intercalating agent is a molecule which is very, very flat, planar, and it slips into the DNA and can disrupt the DNA and kill the cell.
How did you know how effective it was? Was there like certain things was there like a dye, for example, you put into the body to be able to observe what had happened of your intervention?
So once the molecules were synthesized, typically they were very multi-step processes. So for example, 7, 8, 9, 10 steps to synthesize. So you end up with a very, very tiny amount of your target molecule at the end. So we do ID50 and other similar analyses where you measure its efficacy against tumour cell lines. Then if its efficacy is regarded as being kind of potent enough, then it would get sent through to the US for further testing.
And did some of your work get sent over to the US for testing?
Yes. However, I finished my PhD and never found out whether any of those molecules actually ever made it through the rigorous pathway through to an actual anti-cancer drug. I suspect not.
What was the major highlight of your PhD for you?
Yeah, as I mentioned, I was very, very shy growing up. It was probably during my PhD that I started to get some confidence and started to come out of my shell, make more social interactions at university, etc. At that stage, during my PhD, I started studying Japanese, as I mentioned, and I became very passionate about studying that language. So by the time I reached the end of the PhD, I actually wanted to have a bit of a break from science. So I enrolled in a course in Japan, and I went over to live in Japan. I meant to go for 1 year, but it ended up turning into 3 years. So I actually didn't take the natural and normal next step of a science career. I ended up not doing science for the next 3 years after my PhD and being an English teacher.
That's fantastic. Did you have a particular subject that you're interested in teaching? Was it an English teacher as the English language, or did you also have the opportunity to teach like other parts of science or arts or other interests?
Yeah, my main purpose for going to Japan was to immerse myself and learn the language. So I lived with a host family for 1.5 years of that 3 years, so complete immersion. And I was an English teacher in 14 junior high schools for the first 2 years. So the way it works is you rotate through a different junior high school each day and you rotate through them all. So constantly new students that you're teaching. And then the final year was 4 senior high schools. That was all about not so much the academic aspects of teaching grammar, but actually helping the students to enjoy learning English. Over in Japan, at that time anyway, the English teaching was very much based around learning the grammar. So the opportunity for the foreign teacher to come in was all about kind of getting excitement and energy and playing games in English with the students, etc. So I really enjoyed that aspect, and that also really helped my confidence as well.
Can you reminisce and think of any fun games that you used to do? Did you base them on games you knew in New Zealand to help engagement?
Depending on the class, you had different levels of English ability, and between different schools, even different levels of ability. So I would tend to tailor the games to that group of students. But yes, I'd use common games from New Zealand, such as English bingo and other such games. I'd also tend to do a self-introduction in English with some kind of interesting and sometimes humorous points that would engage the students. I used to pass around a little jar of Marmite the students could taste, which got some interesting reactions.
So you spent 3 years then over in Japan. What was the trigger that brought you back into science? Or did you not go back into science after that 3 years?
So I was on a program in Japan called the JET program, which is run by the Japanese Ministry of Education. At that stage, the maximum you could do that program for was 3 years. I did get offered a job by the local regional education department and also by a private high school. So I could have stayed in Japan, which was very tempting because I was loving it over there. However, I had also really enjoyed my PhD. I really love science, so I thought I'd better see if I could come back to New Zealand. And I knew that 3 years was a long time to be out of science, and it would probably be best to look for something in that area. So I came back to New Zealand at that point. Couldn't straight away get a job back in science though, so I actually ended up teaching at high school for 2 years at Papanui High School in Christchurch.
Fantastic. And that was English as well?
No, I was teaching. So to go back a couple of steps, while I was doing my PhD, writing it up, I also went to Teachers College to help me with my preparation for Japan. So I was a qualified teacher as well. So I taught chemistry, taught general science, a little bit of French, taught some Japanese as well, and also ESOL, English for speakers of other languages.
That's a really interesting journey, having these parallel kind of interests of science and then teaching and arts all coming together, sort of woven in. So what was the next big break then, other than Papanui High School? Is that when you came back into science?
Yes, I was very fortunate. There was a job that got advertised at Lincoln with an organisation called Kinesis, which had been the former wool research organisation of New Zealand. So they were a very wool science-focused organisation. And my mother actually found the ad and showed it to me because I was very immersed in my teaching and learning, getting experience teaching. And I looked at it and it really appealed to me. It was in wool science. So I decided to apply for that. And I was very fortunate to get that job. And that's how I started in my science career post-PhD.
Interesting. Wool science.
Yes. Completely different to what I had studied at university.
It was. So it wasn't to do with the chemistry of the wool, or what was it about?
It was a chemistry-based position. There was several wool projects that they were working on at the time in the fundamental science area. I should point out at that stage, wool science was a much bigger area in terms of research than it has currently become. And in particular, I was employed to work on a project that was trying to unravel why wool yellows. So the photo-yellowing of wool.
And why is it important to know why it yellows?
So for wool products, one of the differences they have with synthetic products is the tendency to discolour under UV radiation. And for example, that could be photo-bleaching or photo-yellowing. So photo-bleaching when you might have a carpet in your house and the light is coming through glass. Or photo-yellowing can occur on the actual sheep, and when the wool is scoured or cleaned, that yellow can't be cleaned out. There's yellow that can be cleaned out, which is called scourable yellowing, and there's yellow which can't be cleaned, which is non-scourable. When you look at the wool when it first comes off the sheep, you don't know how much of those different yellows there are.
Is this still a phenomenon that occurs today?
Yes, it's not in the sense of a bright yellow. This is more of a subtle discoloration away from white. But it also occurs in humans. If you observe people who have very white hair, perhaps in more advanced stages of life, it would sometimes go yellow based on the UV exposure, which I believe is one of the reasons why sometimes people do different colours and rinses in later stages of life.
That's fascinating, isn't it? How long was that for, the wool research?
That was a year-by-year project. It was funded by Australia Wool Innovation. This is a major issue over in Australia, as well as New Zealand. And the research went very, very well. I got myself completely immersed in it. It was very chemistry-focused, trying to understand the actual molecular basis for the discoloration, which was completely unknown at that point. But people have been working on it for decades, and it was still a mystery. But we were able to unravel it and essentially make a bit of a breakthrough in the area within the first year and a half that I was working on the project.
Reflecting back, was this part of you trying to carve your niche of where your science research was going to go?
Not really. I was just having fun. So because it was quite a different area, we basically had a proteomics lab, so a whole lot of mass spectrometers. So we'd take wool that had been irradiated at different degrees, so different levels of yellow. Then we'd enzymatically break it up and then fractionate it based on the colour, based on the absorbance at 400 nanometers, and then put it on the mass spec. And essentially, then you end up with a whole lot of peptides. So in mass spectrometry, you have MS, mass spectrometry, and MSMS, which is where you take the ion in the mass spectrometer and you smash it up into little pieces. So my job was to analyse all the fragments of those smashed up peptides and work out what was going on, which I absolutely loved because I loved the analytical aspect of it. All of these unknown fragments, because we were looking for a series of unknown modifications, so not in the literature to that point. So the whole discovery aspect of it, I was very excited about. I wasn't thinking about it from a career perspective at all.
So every day you go into work and you try and find out these new discoveries. That's really fascinating.
Yeah, what I'd do is I'd print off all these MSMS spectra, take them home, and while I was watching TV at home, I'd be just doing de novo sequencing of them with a pencil.
So I guess what led on to then? So at some point in the career, you moved from doing science, more research, and then transitioning more into leading. What point was that?
That was nearly straight away when I got back into science as well. I was called into the chief executive's office pretty early in my career, probably after about 2 years. This is my career back in science, and asked if I would be the equivalent of a group leader, so looking after about 30 or so scientists, and there were various reasons for that. I initially turned it down because I didn't think I could do that kind of thing very well, but then he called me back into his office a couple of days later and asked if I'd reconsider. I reconsidered and decided I'd give it a go. So quite early on, I ended up having some leadership aspects to the science I was doing too.
And during that role, did you have any inklings that the games that you'd use in Japan, would they be useful to bring across into building a strong team of scientists under your leadership?
Yeah, not so much the games, but I learned a lot from my experience in Japan, not just gaining confidence, but around the things that are sometimes called softer skills or the EQ-related things, I believe. So particularly when you're learning a language in a foreign country, you've got to pick up on body language, signals, etc., and particularly in the Japanese culture and Japanese way of communication, which is very indirect. And I found that so useful in a New Zealand work setting where you have meetings about different things and being able to kind of harmonise, work out points of mutual interest. It's really helped with working with different companies and different organisations as well. So I think those kind of aspects I really attribute to that time in Japan and what I learnt there. So the non-science specific things, which are so important in any job these days.
Could you speak a bit more about your time at Ag Research? Was it in a similar capacity leading at Ag Research as well?
I went through multiple steps at Ag Research. So I started off with this role I mentioned earlier. Then I believe it was 2007. We got integrated into Ag Research at that point. So the organisation Kinesis I was with got integrated and became part of Ag Research, which opened up massive opportunities for a proteomics-based team. And we ended up applying our protein chemistry and proteomics and structural biology capabilities broadly across a whole range of new application areas, particularly food science. Then later on, I think it was 2013, I was asked if I'd consider being a group leader within Ag Research. So I ended up leading the food and bio-based products group within Ag Research, which had 6 teams based around different campuses.
What is a bio-based product?
So a bio-based product is a biologically derived product, and one way to conceptualize it is if you think of food production systems, for example, making kiwi fruit, apples, milk, etc., you have your primary product, which is a food in those cases. Bio-based products are typically those products which are non-food, things that come out of that system. So bio-based materials that could be alternatives to synthetic plastics, for example.
What drove your decision to come across to plant and food research?
It was a very, very tough decision. I've never been very good at thinking out into the future from a career perspective at all. I was really enjoying my time at Ag Research, but I really felt I'd like to experience something different and learn new things. At that stage, David Hughes was Chief Executive of Plant and Food, and I'd worked with him for many years on different boards of different organisations, and I really respected his style of chairing and leading meetings and the way he communicated, etc. So that was one factor in the decision as well. But yeah, it was very, very tough. It was very, very emotional leaving Ag Research.
Reflecting back on your career up to that point, it comes across to me as in like maybe you followed your nose more than finding opportunities that suited what you wanted to go for. Could you reflect a bit on that, please?
That's exactly right. I've never been someone who's really planned out a career in a traditional sense. So kind of just considering and taking opportunities as they come up. I always just believe in really, really doing the very best at what you're doing right now and enjoy it. And that's kind of the philosophical approach I've had rather than planning things out.
And that's a fantastic advice to give to early career researchers coming through, and that there's many different ways and on your journey could be to becoming something that you don't even know where you'd be. I mean, when you were younger, did you think that this would be the path you would take? Like, did you have even a vague notion that you wanted to be like in the position you are now? Absolutely
not. A younger version of me wouldn't even have considered or conceptualized this as an option. In fact, in my early years, I really wanted to be an author and write novels. That was actually my dream. I've ended up writing a lot of science papers and science chapters and things like that. So I feel partially fulfilled with having achieved those. But no, I never conceptualized actually, first of all, even becoming a hardcore scientist, and then secondly, from that, kind of moving on into the management and leadership of science.
Mm-hmm. I suppose there is opportunities to leverage when doing science communication to having more of a light-hearted approach to communicating. It doesn't have to be so dry as it would be in a scientific paper. Have you managed to retain those opportunities in delivering science through your teaching skills, and how does that look now?
Yeah, I have adjunct associate professor role at Lincoln and an adjunct professor role at the University of Canterbury. So I've done a little bit of teaching and lecturing associated with other professors' courses, but not in recent history. Just touching on that point, that's one of the things I found very frustrating in science when I came to write my thesis was I from a kind of wanting to be a novelist point of view, you want to use all this creative language, lots of excellent adjectives and power verbs and things. And then I was told by my supervisors for my PhD, we must use consistent, precise language all the time. And if you use that word that time, you must use it again the next time. So I found writing the first papers very difficult from that perspective. But yeah, once you get used to it, it's all good.
Yeah, it's very interesting going into the sort of specific language of each field.
Yes.
Listening or reading geography papers is a very different style of writing and takes your head to get around that too. Could you speak a bit more about your role currently at Plant and Food Research? What does that involve every day to day? And then maybe go on to what do you want to see and what do you want to achieve and get out of this role?
So my current role has two key aspects, one of which is overseeing our externally funded projects. And that's a very wide range of different types of projects, from industry good body funded projects, government funded projects, commercial, international and domestic. Then the other aspect of the role is overseeing the bioprotection and sustainable production portfolios within plant and food. So one aspect of the role cuts across all the areas of science, the other one is specific to those two portfolios. It's quite hard to describe on a day-to-day basis, this role. It's so much variation week to week. There's a lot of aspects of talking with people, helping or trying to understand different areas of science, connecting that through to commercial customers, for example. There's strategic discussions. There's a whole wide range of different aspects to the role.
I had one thread that I'd like to pull on, which is where do you think the role of scientists are in society? Like, you manage a whole load of internal and external projects. Where do you think plant and food science fits in with that general philosophy?
Yeah, it's a really interesting question, and there's many different aspects to that. One aspect, I think, is that of trust. And coming from a scientist's perspective, we want to be trusted and to do really, really robust, good quality research, clearly. In the world at large, however, there's quite a variation in the levels of trust with science, or the concept of science generally. So I think a really important role for us into the future is to make sure we do our best to build and maintain that trust. So from an organisation like Plants and Food, that means doing, very good quality, robust research in an independent way that's not seen to be influenced by either political or other kind of factors, but is seen to be truly as a trusted thinker, as it were, and a source of kind of independent, objective information.
Given that science can be somewhat subjective in the methods we use to find information, which range from what we do in the laboratory to the statistical analysis that are performed, how can we best communicate this uncertainty given the body of knowledge within the entire system?
Yeah, that's one of the challenges in science, I think, is to be able to tell our stories to different audiences at different levels of detail in different ways, being able to contextualize it for people. I think for me the key thing is honesty, but trying to contextualize it for the audience. So trying our best to talk about that uncertainty that's in science, not try to over-represent what we're doing or what our results are, but contextualizing it. There's a tendency to sometimes go into too much detail for certain audiences, particularly if we're very passionate about the subject, we're passionate about the information. I think increasingly that role of communicating science is going to be important in building the trust that we just referred to, but also in really being able to make transformation into the real world with science. We do need increasingly to be able to communicate it well.
And that is a soft skill that is, I think, beneficial to everyone trying to communicate science in this modern world. Looking back at your transition between doing the scientific research as a higher proportion of your workload to now where you've transitioned more into management, is there any advice that you could give to people looking to make that transition as well? Yes, it can be quite a difficult transition to make. Probably one of the best bits of advice that I received was to challenge all your own assumptions. So as humans, we tend to, as we progress through different stages of our life, you can assume that what you learned or assumed from your previous role or what you were doing just automatically can be transitioned through to the new thing. I'm probably not describing it very well, but basically that concept of constantly being ready to learn, take on new things, get feedback and listen to that feedback, I think is really, really important, particularly for this kind of transition. So another way to put it, what made you successful previously doesn't translate automatically into success in the new role. And looking forward to the future, where do you see that your path, I know it's kind of following your nose mostly to this point, but if you had to provide an indication of where you'd like to go, what change you'd like to make in the world or in the science system as it currently is, what would those points be? I'm very passionate
about people and science, and that's what really motivates me around my current role. So the people aspect of science, I believe, is absolutely critical. I think science has changed over the years. There might have been that traditional view of the lone scientist making an amazing discovery and transforming the world. Modern science is so much about connecting with other people, collaborating, sharing ideas, building on ideas, so that how people behave and interact with each other is so critical to actually do both good science and also making a real impact in the world. And it does make me sad. I mean, generally, just an observation about the global science system. Sometimes, egos and personalities can get in the way of that really happening smoothly. So I'm very passionate about that people aspect of science and how we can kind of empower and really fire up people. So we're doing good science, we're enjoying the science we're doing, we're enjoying the work we're doing, but we're treating each other respectfully as we do it.
Thank you, Jolon. This has been a fantastic conversation, and it's been really interesting to hear your journey from science into management.
Thank you.
And thank you out there for listening. If you want to hear more of our podcasts, please check out our Sigest podcast page or subscribe to our channel in iTunes or your favourite podcast provider. And don't forget to post a review and rate this podcast. Matua, and until next time.